Welcome to this presentation on gene expression profiling based on high-throughput sequencing technology and its application in disease diagnosis. This research was conducted by Yuanbo and published in Volume 5 Number 1, 2025, January to June 2025. Today, we will explore the key findings and contributions of this important work. Let's begin by understanding the problem this research addresses. Pulmonary tuberculosis, often called TB, is a serious lung infection that can be complicated by other health issues. This can lead to drug-resistant strains of the tuberculosis bacteria, making it harder to identify and treat the disease effectively. Traditional methods for diagnosing TB have limitations, prompting researchers to explore new approaches. To begin, researchers collected blood samples from three groups of volunteers: individuals diagnosed with pulmonary tuberculosis, PTB, those with latent tuberculosis infection, LTBI, and a healthy control group, HC, peripheral blood mononuclear cells, PBMCs, which are important immune cells, were carefully isolated from these samples using a process called density gradient centrifugation. This method effectively separates the different types of blood cells. The high-throughput sequencing generated millions of tags, which are short pieces of genetic information. These raw tags were meticulously processed to remove any impurities and low-quality data, resulting in clean tags. These clean tags were then aligned to the human reference genome to identify the genes they originated from. The findings revealed a complex picture of gene activity changes in pulmonary tuberculosis. A large number of genes were found to be either upregulated, increased activity, or downregulated, decreased activity, when comparing patients with TB to healthy individuals. Interestingly, while more genes were initially found to be upregulated, the downregulated genes often showed much larger fold changes, suggesting a more dramatic impact on their normal function. This research successfully employed high-throughput sequencing technology to analyze gene expression differences in peripheral blood mononuclear cells between individuals with pulmonary tuberculosis and healthy controls. Study identified several genes that exhibit significant changes in their activity levels in the presence of TB.